Hello friends, we are Macarena Céspedes and Sofía Cesari, and in this section of Genetic and Bioinformatics, we will talk about a very interesting scientific topic that addresses the origin of new species, that is, speciation. But what is speciation? Speciation is the source of the diversity of life. In fact, it is defined as a process by which a population of one species gives rise to another species. Can you imagine this process? First of all, we define species in broad terms as natural populations that share a series of distinctive features and which are capable of reproducing each other. Geneticist Georgi Shan of BGI Genome Research Center in Chechen, China, delivered a very detailed bird family tree. There we see a clear picture of how modern birds originate and evolve over time. Their analysis focused on a group called neobirds, which includes almost all of more than 10,000 species of birds. Species include penguins, hawks, eagles, woodpeckers, owls, vultures, pelicans, cormorants, ducks, chickens, and turkeys, among many other species. This relationship between different species of birds also occurs in the aquatic world, terrestrial among others. In short, everything that is and is defined as speciation is what we have said before, but we must investigate more thoroughly why and how this evolutionary process occurs. How do the different species originate within a single population?
Then, how are new species formed? The investigation related to the formation of new species or speciation is rich in the current historical debate. Evolution can be defined as a new variation in populations through mutation and genetic recombination. These new species are formed when individuals of divergent populations cannot be recognized as potential partners, if the mating opportunity is limited, or by the difference of reproductive schemes. With this, we can say that there exist viable and unviable individuals, from where, in a hybrid viable, an unviable zygote can exist with sterile descendants, which are considered failures in viable hybrids. The way to prove this viability is with pre- and post-zygote barriers, which will determine how reproductively isolated is one species with another, or, in other words, to what point speciation continues. A major area of debate among speciation biologists is the geographic context in which it occurs. Three types of speciation by geography exist, which are allopatric, parapatric, and pure sympatry. The first one is speciation by geographical isolation. The second one refers to populations that are situated in geographic proximity to one another, usually with abutting but non-overlapping ranges, and the third one by overlap populations. On the other hand, there is the ecological speciation, which is defined as the process by which barriers to gene flow evolve between populations as a result of ecological-based divergent selection between environments. This process makes some simple predictions. For example, ecologically divergent pairs of populations should exhibit greater reproductive isolation than ecologically similar pairs of populations of similar age. It is only in the past 5 years that studies of speciation have truly entered the molecular era. Recent molecular analysis of a handful of genes that are involved in maintaining reproductive isolation between species, speciation genes, have provided some striking insights. In particular, it seems that despite being strongly influenced by a positive selection, speciation genes are often non-essential, having functions that are loosely coupled to be reproductive isolation. Molecular studies might also resolve the long-running debate on the relative importance of allopatric and parapatric modes of speciation.
At present, how have the relationships between species been related and studied? With the help of new technologies applied to biology and computer science, accessible software such as Mega and its various versions have been developed. In the classroom, we have seen Mega 6, which analyzes the DNA and protein sequence of species and population data. In addition, allows the construction of molecular evolutionary trees scale at the time. When constructing a bisection tree, the user must provide the program with a file containing a sequences, alignment, and another containing a phylogeny. That is the relationship between tags of groups of related organisms. After verifying the calibration restriction, the option to display the data in a tree diagram is selected in the program.
Created using Powtoon.